Hi, welcome back. Today we're going to talk about Chapter 6 in your Concepts of Biology textbook called Reproduction at the Cellular Level. Let's get started. I wanted to begin this chapter with a story that is not in your textbook, but I find is very relevant to what we're talking about today. There was this lady in the 1950s named Henrietta Lacks. She had cervical cancer and she sought treatment at her local hospital. Now, she and her family were not educated. They lived in rural Maryland and they didn't have much contact with authority figures, if you will. So, when Henrietta went to this hospital, the hospital was actually segregated because you have to remember this is the early 1950s and segregation was still a big happening at the time. And so when she went to the hospital was actually segregated. So she went to the black side and the white people were separated and went to their white side. And so the doctor that saw her approached her as he would at the time of any black person that he dealt with. In his eyes, they were a lesser being, and so he could do whatever he wanted because he didn't need permission from her. He was the authority figure in the scenario, and so he did what he wanted. And in this case, he was doing what he wanted in reference to his own research. So when he took cells from Henrietta, he determined that she had cervical cancer and it was rapidly growing and metastasizing to other parts of her body. And he saw something unusual in her cells, and so he did biopsies and he kept those cells. And in the meantime, he provided treatment to Henrietta, and at the time, again, they didn't know that this was not a proper way to treat cervical cancer. They radiated her stomach, so it would go right through to her cervix. And she would come back time and again for treatments, and she would feel better for a little while, and then she would feel really sick. And so the radiation was actually making things worse in her body because, as we know now, we can only tolerate a very small amount of radiation before it starts acting negatively upon us. So that sort of sped up her death in a way. Meanwhile, the doctor's noticing that her cells have this uncanny ability to replicate outside of a human host. And for decades, even up to 100 years, scientists had been trying to figure out how to do this. And they accomplished this several times throughout these studies. The cells would live outside the body only for a few days and then they would die. But Henrietta's cells would not only grow, they were very prolific. They would grow time and again without a human host, without being inside a body, just inside a petri dish or in a test tube, and they would grow incredibly rapidly. And so they knew they had something really amazing here. And at the time, they sent cells all across the world for people to study. What's unusual about this is that these cells are actually still dividing today. These cells were kept in a warehouse for a while because they had so many, they had to build a facility to house them. And scientists from all over the world will write to the organization that holds them and says, hey, send us some cells we're working on a specific project or study or whatever it is that they want to look at. And so they would get cells for free. Now I think they have to pay for them because now it's become such a big deal. But throughout this process, one of the researchers that obtained these cells used them to fabricate the very first polio vaccine. And so these cells are incredibly useful. And if you've ever had a transfusion or worked in a facility that was using cells for their study, chances are that you've used these Henrietta Lacks cells. And they're called HeLa cells, based off of her name, HE for Henrietta and LA for Lacks. So HeLa cells. And they're just incredible and a useful tool that we still use today. One thing notable about her story is that long after she died, a young lady came to her family, her daughter especially, and said, "I'm writing a book about your mom and all the ethical challenges that she faced and that you're facing, along with the scientific part of it." And from this particular story, we now also have laws in place to protect patients. Henrietta never knew that the doctor had taken her cells and used them. And he used them for gain, as well as many, many, many other people that use them at this time. And her family didn't know about it until this reporter lady came and started asking questions so that she could write her book. And they were outraged, of course. And of course, their first instinct was to say they deserve part of the profit that's been happening. So a giant lawsuit ensued, and it was actually just settled on, I think, in 2012 or 2013. And it was found in favor of the family, but they were not awarded any monetary settlement. They were given recognition that her family member has allowed scientists all over the world, all over these decades, to come up with different types of vaccines and cellular transference processes and things like that. And so her cells are invaluable, basically. But there's a museum, I believe, and there was something else that came of it for them. We'd have to look it up in the settlement, but the book that the reporter wrote has become quite famous, and it's called The Immortal Life of Henrietta Lacks, Lacks. And you can find it at our local library. You can get it through Amazon and Barnes and Nobles, and any bookstore, any library is going to have it. And it's a very short book, and it's a very good read. I highly recommend it, and I liked it so much that, and found it very relevant to what we're talking about this week, that it's your very first extra credit opportunity. So go to your modules, scroll all the way down to extra credit, read the extra credit introduction, and then read the extra credit about the HeLa cells, and you'll be able to get extra credit for reading this book for this term. Okay, now I promise to move on. So this picture should take you back to our very first week when we were talking about atoms and elements in single cells. So the picture A is showing you two different cells. These are actually one cell dividing into two, but remember that two atoms fused together would become a molecule, which is what you see in letter B, is a bunch of cells all grouped together, and those are called molecules. And then, of course, molecules form to make cells, and then cells form to make tissues. And so then we get letter C, which is your final organism. So B actually is your groups of cells, and A is one cell singular that's dividing into two. Right, so this is a picture of a karyotype, and in the little box there to the lower left, the little rainbow colored squiggly lines, those are called chromosomes. And a karyotype is a tool that a geneticist will use to quickly glance at a person's genetic makeup, their human genome, if you will. And you can see they're numbered 1 through 22, and then the last one in the lower right is an X. The chromosomes that are in the numbered figures, so if you look at number 1, it has 2 identical yellow lines. And then same with 2 and 3 and 4 and 5. Now number 2 is a little funny looking. You wouldn't think that they're identical because one is curved like that, but if you straightened it out, it would look exactly like its right-hand counterpart. And so these are chromosomes that are paired together because they're exactly the same, and they are numbered 1 through 22. These are called your autosomal chromosomes because they are not your sex chromosomes. Your sex chromosomes come with chromosome number 23. Now, they didn't number it to 23 in this case, they just gave you the letter X, which is a female. If you remember, females have XX chromosomes and males have XY chromosomes. So sometimes you might see, if this was a male, you would see a Y and an X, and then those two green chromosomes would be separated. One would be over Y, one would be over an X. So you'll see the sex chromosomes written in different ways here. But in here, we have a female because there's 2 chromosomes that are the same over the letter X. So we have a female human karyotype here. And you'll notice in the text it says DNA plus proteins equals chromosomes. And a human genome consists of 46 chromosomes. Those are the individual ones that you see like in the rainbow colored squiggly lines there. But when they pair up in this manner, we have 23 pairs, and we have 2 sex chromosomes. Okay, and then a genome, just the definition of a genome, is a complete complement of DNA. This is just another type of karyotype that we're looking at. It's actually the same one as previously, but just blown up, and it's featuring numbers 4 and 5. And you can see how the 2 green ones are exactly the same and the 2 purple ones are exactly the same, but the green ones are not exactly the same to the purple ones. So we can say that chromosomes number 4 are homologous to each other, and chromosomes 5 are homologous to each other. But chromosomes 4 are not homologous to chromosomes 5, and of course vice versa. And so we have 2 pair for each number because one comes from our mother and one comes from our father. So we always have 2. Okay? And that's in a normal, healthy human. Homologous means that they are the same, so they have the same length, they have the same nucleotide sequence, which we'll talk more about in a little while, and then they have the same genes at the same loci. And you can kind of see that on chromosomes 5. See the little darker band towards the top? Those would be your genes. Okay, and that would be, let's say, that's the gene for hair color. Well, that gene for hair color would appear at the same spot on the chromosomes every single time. All right, we only have errors in chromosomes when those genes start appearing in different places on that long little strip of chromosome. Okay. Remember, homo means the same, so these are homologous. These are the same. Remember in the beginning of the semester how I talked about homeostasis, and the sole goal of an organism is to find a mate and pass on its genes to its offspring so that hopefully they can grow up and find a mate and do the same. This is the process that happens with that. We are looking for a mate so that we can reproduce and have our genetic material passed on. Genetic material, once it's passed on and the organism starts to grow, each cell will divide into and continue to grow until we have an organism. Well, each cell receives a copy of the parental material, of the parent's genetic makeup. So half of our genetic makeup will come from our father and half will come from our mother. And so every descendant cell receives a full complement of chromosomes and some of the cytoplasm. If you remember, the cytoplasm is sort of that hair gel type material that all the organelles inside the cell kind of sit within and can move within it as well. So before the cell cytoplasm divides, that is, it must replicate all those chromosomes, everything we saw on that karyotype. It's got to duplicate all of those before the cell divides. That way, once the cell divides, a full complement of chromosomes can go into one daughter cell, and then the other set can go in the other daughter cell. So the cell that is original, that's getting ready to divide, that is called the parent cell, and it has a full set of chromosomes in it. And then once it goes through the division process that we'll outline in just a minute, once it goes through that division process, it makes two daughter cells, and each daughter cell has a full chromosome complement from its parent. So mitosis and meiosis are two different ways that cells can divide. Okay, mitosis is usually connected to asexual reproduction, where you don't need a mother and a father to reproduce. You only need to be one cell and you can divide all by yourself. Okay, that's asexual reproduction. So one cell gains two daughter cells without a partner, basically. Meiosis is the opposite. This is what most organisms we're familiar with will undergo to divide. And so meiosis is used in sexual reproduction. There's two parents contributing genes to the offspring. This chapter we're only going to talk about mitosis, and next chapter we'll talk about meiosis. They can easily be confused, so we're going to separate them down into two separate chapters so you can just focus on one at a time. But I wanted to present them together so you could see the difference easily. One is asexual reproduction, the other is sexual. You'll see in your book that there's this pie graph looking illustration, and this is going to show you a brief pictorial of how the cell divides and the cycle that it goes through. And there's three phases: interphase, mitosis, and cytoplasmic division. And so the cell cycle is just the process that a cell undergoes when it's dividing. We call it the cell cycle. So interphase is the first phase that a cell undergoes when it's getting ready to replicate itself and pass on its material to its daughter cells. And most of the cell's activities occur during interphase, and it's the phase that occurs between mitotic divisions, when a cell grows, doubles its number of cytoplasmic components, and replicates its DNA. There's three stages: the G1 or gap 1. We usually call it G1, but you might hear first interval as well. So the G1 phase is where all the growth happens for a DNA replication, and most cells will stay in this G1 phase. It's not until it enters the S phase of interphase where it starts to divide. And so in order to divide, it must duplicate its DNA before it can split into two. So the S phase is where DNA replication happens. We're making a copy of our DNA. G2 is the second interval or the second gap phase, and this is where the cell gets ready to divide. So interphase happens before the mitotic process, and it's G1, S, and G2. Here's that graph I was telling you about. It looks like a pie graph. You'll often see mitosis depicted like this, and so this shows you all the three phases. It's got interphase where it has G1, S, and G2, and then it shows you mitosis and then cytokinesis. It shows the mitotic phase is making those two daughter cells along with the cytokinesis. The first cell, or excuse me, first G1 is where the cell growth happens, and then if it moves into S phase, that's where DNA replicates or DNA synthesis, and then we have the last stage where the cell grows in G2 phase. This is just another way to think about this particular picture. In the G phase, it's going to show you that the G1 phase here, where cells start to grow, don't actively divide in this stage. That happens later on. And so the G1 phase is where many cells just hang out and stay. They never make it to the replication stage. And they don't divide. They just grow, and then once they reach their maximum growth, they just hang out in this particular phase. So how does a cell know when to move on to the next phase, when it's allowed to move on to the next phase, or when it's being kept in the G1 phase, for example? There's specific controls that the cell has that lets it know, okay, we're done with this phase, let's move on and get the job done. So if we're in G1, there's a checkpoint. So picture yourself in Europe and you're walking up to the checkpoint between Germany and France. And so you walk up there, you have a checkpoint, they stop you and say, hey, may I see your passport? Do you have a right to be here? Do you have a right to be passed on? And should I let you pass on? And so once you're given the okay, then you can move through the barrier from Germany and move into France. And I use that example because it's what I think of every time I hear checkpoint. When I lived in Germany, that exact thing happened to me. I was out joyriding, trying to learn the countryside and enjoy the beauty of it, and I accidentally came up to the border, and I didn't have my passport on me, and they wouldn't let me go through. And so every time I hear the word checkpoint, that's what I think of. Anyway, the cells will have their own checkpoints. So if you're in the G1 phase, you've grown, you've done all that you can there. Now you come to this checkpoint and you say, hey, can I move into the S phase, please? And it will say yay or nay. If the checkpoint is active, it will say, yes, you are completely grown the way we need you to. Here's your passport to go through. Okay, so there's a checkpoint between G1 and S phase. There is not a checkpoint between S phase and G2. Often, once the cell replicates, it moves immediately into the G2 phase. Okay, so it doesn't spend much time in that S phase once it's duplicated. So there's no need for a checkpoint there, because once it divides, it has to move into that G phase so that it can move on into the mitosis phase. Okay, it can't hang out. You're not allowed to hang out in S or G2. All right, so there's a checkpoint between G1 and S, and then there's a checkpoint between G2 and mitosis. So once you get to G2, you've replicated, the two of you are hanging out here in G2, you've got to get permission to move into mitosis phase. And again, the cell, these checkpoints will say yes or no. Okay, if they're damaged or not complete, they will either be destroyed or they'll just be left there to hang out. But usually they're destroyed. on occasion they can be repaired, but it just depends on what's happening at the time in that cell, whether or not it will be repaired. If it gets repaired, then it can get permission to move through. So we have checkpoint between G1 and S and G2 and mitosis. And once again, here's that depiction. You can see the little squares now. If you look at the end of G1, there's a square. That's your checkpoint. And if you're allowed in, you get to go to the S phase and duplicate, and where you'll immediately move on to the G2 phase, where you can grow a little more if you need to. But then there's another checkpoint there at the end of G2, and it will say, hey, yes or no, can I go through? And it'll say hopefully yes, you can, you're 100% complete, and we'll move into mitosis. And then you'll notice there's one last checkpoint, and that is between the end of mitosis and the cytokinesis. Okay, and that's again going to say yes or no, you can pass. Once cytokinesis happens, then you've got the two daughter cells forming. And so you want to make sure everything's really cool and in its place and proper and healthy before you allow it to move into two daughter cells. Because once that happens and they start to divide, that defect could be carried out from adult to daughter all the way through the genetic line. So we want to make sure we get everything correct in a healthy way in the correct positions before we let the cytokinesis happen. So let's talk about this idea of maintaining the chromosomal number. So remember, think back a few slides to that karyotype picture. It had numbers 1 through 22 plus the X chromosome, and so that was a female. In order for all of that genetic information, 1 through 23, or 22 in the X, however you want to think of it, in order for all of that to get passed on to the daughter cells, we have to duplicate the cell. But if you were to take one single cell with all 23 chromosomes in it and you divide it in half, then only half of that information is going to be in the daughter cells and half will be in the other daughter cell. And so now you have dysfunction. Every time that cell divides, it's going to lose part of its chromosomal numbers. So if you multiply your chromosomes, all those chromosomes 1 through 23, if you multiply them first, then you have 2 complete copies, and then when the cell divides, you've got 1 complete copy in one daughter cell and a second copy in the second daughter cell. Otherwise, if you divided prior to if you divided before you replicated, you're going to have 11.5 chromosomes in each daughter cell. Whereas if you multiply it first, then you have 23 chromosomes in one and 23 chromosomes in the other. Okay. Also note that we have more terminology. This chapter gets into a lot of terminology for things that seem the same. And I know that can get confusing and obnoxious, but they're necessary. So when you're in the G2 phase, you've already duplicated yourself, and we're getting ready to split once we enter into mitosis. And so it says here, it talks about centromeres and chromatids, sister chromatids. So here's a visual. And if you could see me, I'm talking with my hands so that you can tell exactly what we're talking about. But picture one single chromosome, so it looks like a little line, right? And then you have a second one next to it, so it looks like the letter 11, we'll just say. Those two are going to attach to each other in the middle. of one another. So it's like you pinch them together, and when you do so, it creates this little X pattern. Okay, because think of it as they're side by side in the letter 11, or excuse me, the number 11. You pinch them together in the middle, and then the ends, the top ends and the bottom ends, can freely flow around, and so they're attached just at the middle, and the tops and the bottoms kind of move around, and then they make this X shape that you've seen before. Okay, so now we're in an X shape. When they're tied together like that in the center is called the centromere. Those two chromosomes that are attached together are called sister chromatids. So once those chromosomes attach at the center by the centromere, they're now called sister chromatids. They're identical to each other. They're sisters, okay? Think of them as twins. When they are pulled apart during mitosis, half will go into one daughter cell, half will go into the other daughter cell. Okay, but in order to do that, they have to be pulled apart. And so mitosis is the process of taking those sister chromatids and pulling them apart, and then dividing the cytoplasm and nuclear envelope so that we end up with two new daughter cells. Okay. Once the cytoplasm divides, they are then said to be two separate cells. Okay, so let's get into the steps of mitosis, and this will all make more sense. This is another picture that is not in your book, but I find that the simplicity of it helps make sense of what's happening during mitosis and how we keep that same chromosomal number through each cell division. And so letter A is showing you a parent cell, and it has one copy of chromosome from your father and one from your mother. And as you notice by G2, so somewhere in here in between the first and second cell, we reached the S phase, we duplicated, and this is what G2 looks like. Now we're sitting there hanging out, waiting for the mitosis part to happen. So between A and B, we had the S phase. Now B is at the G2 phase. You can see how each parent chromosome was duplicated. Now I have two blue and two pink, so two from the father, two from the mother. And then we go from G2 all the way into mitosis and cytoplasmic division. We end up with two daughter cells here in part C, and you can see how one blue chromosome went into the top cell and one went into the bottom cell, and the same thing with the pink chromosomes. One pink went into the top and one pink went into the bottom. So that's how we maintain that chromosomal number. If we just went from A to two new cells, we just cut the A chromosomes in half, and we did half of the blue in one daughter cell and half of the blue in the other daughter cell, you can see where you would have an incomplete set in each cell, and so you would have major defects in this particular child. Okay, so this is outlining the phases of mitosis and cytokinesis at the end. So if you notice at the very bottom, it's bracketed the very first phase all the way to the end with telophase, and then the last column is your cytokinesis. Okay, so let me point something out to you. Depending on the textbook you're looking at and the professor that you're talking to, the prophase and metaphase columns usually have a phase in between, and that phase is usually called something like this one, prometaphase. Sometimes it can be called second prophase. Sometimes it can be I forget what, there was another name for this, but it will have several different names. The good textbooks, in my opinion, will not separate this out. because it gets confusing, because there are different ways that it can be called, and different people will say different things happen during this phase depending on what we've called it. What I like to do, and what makes the most sense, combine the second column with the first column. So then we end up with prophase, metaphase, anaphase, and telophase. Okay, so for our purposes in this class, we are going to have 4 phases of mitosis. Everything that's happening in the pro-metaphase column here is going to happen in prophase. We're just going to group it together and make one big column called prophase. Okay, so there's a lot happening here, which is probably why they split it up, but for our purposes, we're going to talk about it all together. So we have the first phase, prophase. Prophase is the very first phase of mitosis where you can actually see the chromosomes, the individual chromosomes, those little X patterns that we're used to. Prior to this, during interphase, especially after the S phase where there's chromosomal duplication, the chromosomes are still swimming around kind of as individual spaghetti string. It's just kind of all wild looking. They're not grouped and packaged together yet in this X formation that we're used to. Okay, and so you cannot see with the naked eye the individual chromosomes in interphase. The very first time you can see chromosomes is during prophase, because they've wound up tightly, and they've packaged each other in these X shapes. So the sister chromatids came together in the middle with the centromere, making that X pattern. Now we can see them under a microscope, and we call that prophase. So the very first time you can see chromosomes is during prophase. The other important thing to think about in prophase is that the nuclear envelope is breaking down. And so the nuclear envelope is what envelops your nucleus, right? And so when that breaks down, the contents of the nucleus can now be seen, they can now be approached and taken to be copied. And so that nuclear envelope breaking down starts this whole mitosis process. All right, and then you'll see in the second column here that we're calling prophase still, you can see how it's sort of getting this oblong shape. It's not round, and it's sort of like a football in a way, and you have 2 distinct ends on the left and on the right. The areas that congregate at the point of that, so that little point on the left hand side and on the right hand side, those are centromeres, okay, and they are pulling, or excuse me, they are having attached to them these spindle lines. So you can see how those lines shoot out from the centromeres. They look kind of like spider webs. And then you've got the chromosomes in the middle, or starting to get in the middle. Those spindles are pushing those chromosomes to the middle so that we can get to the metaphase portion of prophase. Okay, so here you see the poles with the spindles, and they're pushing the chromosomes towards the middle. So all of that happens in prophase. In metaphase, the spindles have lined up all those chromosomes exactly in the middle where they need to be. So that's the important thing about metaphase. The chromosomes are now lined up in the middle, and that's the easiest way to recognize the metaphase phase. Notice the spindles are still attached to these chromosomes. That's going to be important for the next phase, which is anaphase. Anaphase has the spindles attached to the chromosomes, and the polar ends, which is that left hand and right hand side we were talking about, start to elongate, and as it does so, the spindles that are attached to each chromosome will pull apart. And so it will pull one chromosome to one side and one chromosome to the other side. Okay, and that's the most important thing about the anaphase, is that they're now separating the chromosomes. In telophase, we pretty much have our two daughter cells, but they're not divided yet into two complete cells. So in telophase, the nuclear envelopes wrap around all the new chromosomes on each side, so you can see the circles in the middle, and the spindles have sort of let go, and we are ready to move into cytokinesis. So mitosis is the prophase, metaphase, anaphase, and telophase. And then you have cytokinesis. Cytokinesis means that the cytoplasm is dividing, and as it does so, it creates two new daughter cells, and they will be separated in this phase. So cytokinesis you can kind of see how it's got that divot at the top and bottom where it's starting to converge at the center, getting ready to pop apart. That area that dips inward on the top and bottom, that's called a cleavage furrow. You're making this cleavage, a furrow, a canyon, however you want to think of it, but it's starting to come together as two separate cells. This is just a quick little mnemonic to help you with the phases of mitosis. Again, we're going to be just considering them 4 phases of mitosis. So we have prophase, metaphase, anaphase, and telophase. And so you can think of it in this mnemonic of Paul met Anna at the theater, or whatever works for you. But that's one that the textbook usually uses. And then we're just going to outline each phase of mitosis again. So the prophase, the chromosomes condense and spindles form. Again, you can see chromosomes for the very first time here. Nuclear envelope breaks up, and those spindle microtubules attach to the chromosomes. And then the spindles will move during nuclear division. So mitosis is the process of nuclear division, and cytokinesis is the process of cytoplasmic division. Metaphase is where you can see all of the chromosomes lined up in the center of the cell. So all of those spindles have pushed the chromosomes to the middle there, and they're getting ready to be yanked apart, actually. Alright, and then during anaphase, the microtubules have pulled apart the sister chromatids, so now we've got half on one side of the cell and half on the other side of the cell. But now each side has a complete set of chromosomes, and that again goes back to the S phase of interphase, multiplying prior to division. So here is our last phase of mitosis, which is telophase. And during telophase, the nuclear envelopes start to form around each set of chromosomes. So each set of sister chromosomes that were pulled apart. Now we've got this nucleus starting to appear around both sides. All right, and then the two nuclei form. And so now each set of chromosomes is starting to look like a new cell. So here are some pictorials again that are not in your book that might help you visualize what we've been talking about in mitosis. This picture, the next few pictures actually, will show you what mitosis looks like in a plant cell and in an animal cell. And normally in this class we just sort of overlook the plant cells, but because we're only dealing with animals because we're human medicine. But what you will probably notice is that it's easier to see what's happening in these cells in the plant cell pictorials. Okay, so look at either one, whichever is easiest for you. All right, so this is showing number 1, interphase. Okay, this picture of interphase is cloudy. Not a lot's happening other than we've duplicated our chromosomes and we're waiting to move into mitosis. We're getting ready for that checkpoint to let us through. Okay, so you can see how it's very cloudy and you can't see. You can see there's something under there, but you're not quite sure what that is. Okay. All right, let me point this out. Here, this slide is the very first step in mitosis. This is prophase, but you'll notice that this particular slide, they're calling it early prophase. And the next slide is going to say prophase. So we are going to combine this slide with the next slide and call both of them prophase. Okay? And so what's this showing you is that we've now duplicated our chromosomes. So we have 2 copies of our parent, each parent, 2 copies of the father, 2 copies of the mother, which are shown here. And now we can see them for the very first time during this phase. And you can see that in the plant cell really well. Okay. This is showing you prophase. So again, this is an extension of the slide that we just saw. And this is also why I like these slides, is because the cartoon picture really depicts what's happening in each phase really well. Because if you look at the live slides, the mitosis in the plant and in the animal, sometimes it can be a little difficult to discern what is happening. So I think the cartoon pictures are actually really great. So this is more of prophase. You can see the chromosomes now, but now you can also see the centromeres, or excuse me, the centromeres on either side, the left and right, starting to form, with the spindles starting to be formed and reaching out towards those chromosomes. Okay, they're going to attach to those chromosomes. Now we can move into metaphase. This is the second phase of mitosis. You can see now all the spindles have lined up all sets of chromosomes in the center of the cell. The plant cell actually and the animal cell really depict this very well. The animal cell especially, you can see the centromeres with the spindles, and then the black line is the chromosomes. And so we've got all the chromosomes aligned in that center. Now we get to the fun part. We are in anaphase, and anaphase is where the microtubules are going to take each chromatid and pull them towards the poles. Pull them towards left and right there. Pull them towards each other. Okay, so we're starting to pull apart. And that's anaphase. And you can see again in the plant cell it looks really amazing, and in mitosis now they're separated. Finally, we come to the last phase of mitosis, and this is called telophase. And you can see now in the cartoon picture how we have 2 sets of cells. They're still attached there in the middle. Notice that. That's important, that they're still attached, but there are 2 copies of each chromosome. So there's you've got the mother and the father. You've got complete sets in both. And the animal cell really shows this well, how there's 2 sets here and they're almost completely separated, but they're not quite. They're still attached. Okay, so that's important. It's the end of telophase, but they're not yet daughter cells. Once we move out of mitosis, we've left telophase alone, and now we're into what we call cytoplasmic division or cytokinesis. You'll hear both interchangeably. It just means that we are dividing the cytoplasm into 2, and the end result is going to be 2 new daughter cells. Okay, so each new cell is going to have its own nucleus with 2 chromosomes. We call 2 chromosomes, we call them diploid. Di meaning 2. So now we have 2 chromosomes in each new daughter cell. Okay. And again, it's talking about how the phases of mitosis, the anaphase and telophase, really started to make this cytoplasm converge inward with a ring. We'll talk about the ring in a second, in order to be able to separate the cells. So if you're in the last phases of anaphase and telophase in mitosis, you'll see the cytoplasm start to converge towards the middle. Think of a balloon that you've put a rubber band around the middle of the cell, or excuse me, middle of the balloon, and you've tightened it. Okay, think of that visual. So that rubber band would represent this contractile ring, and it's going to start squishing that cell inward, pulling it tighter and tighter. It makes this cleavage furrow, which is where it's really deep. It looks like a deep V on the top and an inverted V on the bottom. And it really pulls them in. And at the very end, it's going to pinch together one last time, separate into two separate cells. Okay, and of course, each new cell is going to have its own nucleus and genetic information. Here's a pictorial for you. The top is the animal cell, the bottom is plant cell. You can see how we get this cleavage furrow with this contractile ring, and as it tightens more and more, it pops the cells together. It pulls that tightly so that they pop open into two separate cells. And the plant cell doesn't show them separating, but they're now two separate cells as well. Here's a picture that I really like. It shows you the entire cytoplasmic division, or cytokinesis. It shows you the cell in the last phase of mitosis, which is telophase, and it starts to get this contractile ring in the middle, and it starts to pull tighter and tighter, as you can see, until pop, at the end, the contractile ring pinches off, and now you have two cells staring at you because they look like little eyeballs or little eggs. But that's a good picture. I also like the little drawing that they have showing the contractile rings. It's pretty cool. So everything I've talked about till now has been dealing with how we duplicate our chromosomes, our genetic information, and pass them on to each new cell that we create. And each daughter cell then becomes a parent and goes through the whole duplication process and ends up with daughter cells of their own. And that goes on and on and on and on until you have an organism formed, or an organ formed, just depending on what it is. All of that has been in a healthy individual. Now we need to talk about what happens when there are some illnesses or ailments or unhealthiness happening. We like to call them pathologies. Pathologies just indicate that there is something going wrong, something's not healthy or happening here. And there's many different ways or areas that things can go wrong. Basically, at every step along the way, you can have something go wrong, but there are some key areas where if things go wrong, we're in big trouble. So let's talk about the first one. If a checkpoint mechanism fails, the cell may skip interphase altogether and start dividing with no resting period. And so what that means is it's gone through G1 and G2, even G3, or I said G2, G1 and S. So it's gone through G1 phase where it's grown, and S phase where it's duplicated, and it may even still be in G3, I'm sorry, G2, but it's not going to have this little stop period that makes it wait until everything's ready to have it divide. Okay, so if all three of those go and there's no checkpoint at the end, they can keep dividing again and again and again and again till the end of time. And this is what's happened with Henrietta Lacks. Her cells just kept dividing like crazy. She had no checkpoint to make them stop growing or stop duplicating. So hers have spun out of control, and which is why they've been able to duplicate for 60 years. I mean, that's just astounding. And I hope they make the movie of her book, because I think it's fascinating and it kind of illustrates this process of what happens when your genetic material or genetic processes fail. And Oprah apparently was going to make a movie out of this book, which I hope she does. um, but as of yet, we don't have a movie. Anyway, so you can also have singling mechanisms that will make an abnormal cell die, or may tell it to stop moving out of control. So there's some processes that you can say, oh wait, oops, I noticed that we didn't quite stop dividing yet. Let's go ahead and stop, and we'll signal that, and we'll make it stop. But now we've got this abnormal growth. It didn't grow out of control, but we do have this growth happening, and it's we call it a mutation or a neoplasm. So a neoplasm is an accumulation of cells that have lost control over how they grow and divide. Okay, that's what a neoplasm is. Mutations are passed down to every cell from here on out. Um, so let's say we had this abnormal amount of growth. Every cell that gets that genetic information is now going to have this abnormal growth. Mutations are always passed on. It's how we get genetic diversity. So all mutations are not bad. Some mutations are important. They are what make us different from anyone else. So if we did not have mutations, we would all look exactly the same. We would all have the same processes. We would have all the same phenotypes, which is your outward appearance, and nothing would ever be unique. So we need mutations, but of course some are good and some are bad. Another name for a neoplasm is a tumor, and that just means that there's a lump that is forming. Okay, so a tumor has genetic material or cells that have grown out of control, and at some point the lump will stop growing and we see this tumor, and in other cases it won't stop growing and it becomes a malignant issue. So an oncogene is any gene that helps transform a normal cell into a tumor cell. And if you think of the word oncology, this will make sense. Oncology means an abnormal cell or a tumor that is present. And then genes for proteins that promote mitosis are called proto-oncogenes because mutations can turn them into oncogenes. Okay, so now we're able to pass on that tumor material to our offspring, unfortunately. One thing I'd like to mention is I've talked about how mutations are not always a bad thing. Mutations, even if it's a bad one, will be passed down in your genetic information, but just because you have that gene for an ailment or pathology does not mean it's going to manifest itself in something harmful to you. So your outward appearance may not change, your internal processes may not change, but what happens is you have the gene that has the error in it, so you have a potential to have the ailment or pathology manifest in your outward appearance or your self processes. So the gene will be bad, it will be passed on. It may or may not manifest itself in something you can see or feel, but you will still have it. you will also still be passing it on to your children because it's still in your genetic makeup. Okay, remember, mutations get passed on. Um, those children have the potential to have the disease as well. And sometimes when you hear something has skipped a generation, that's how that works. So let's say I have it, I have the gene, and it manifests in a negative way. I pass it on to my child, and they may or may not show it. Let's say they don't show it, but then their children, my grandchildren, have the same genetic mutation, and now they're showing in a pathology for it. Or it could be that we all show the pathologies, or it could show that we all don't show the pathologies, but the gene is still there. And so that's what they're talking about here with the BRCA gene. BRCA 1 and 2, those are breast cancer markers. Those are genes that we look for in familial histories of people that have had breast cancer. So we want to type them and see if their genes are present. We would if you had breast cancer and you have a child, a female child, you would test the female for these genes, and if they show up, child now as an adult would have to decide, do I want to have a mastectomy, a voluntary mastectomy, or do I want to wait and see what happens and see whether or not I get breast cancer myself. So it's an incredible tool to have, to be able to mark these genetic pathologies, these defective genes, and it poses a lot of turmoil in patients because sometimes knowing isn't always better. But in the case of breast cancer genes, I think it's definitely important to know because it's such a huge prevalence in our society. So, you'll get tested for this if you have a familial history of it. If you don't, they probably won't test you because it's an incredibly expensive test to have done, and insurance probably wouldn't pay for it. If you have a familial history, then that tells insurance that you have a direct need to have these genes typed or to look for these genes. And so that's a whole different ball game as far as insurance is concerned. Viruses such as HPV can cause a cell to make proteins that interfere with its own tumor suppressors. So in HPV and the BRCA gene, what's happening here are that the checkpoint genes are inhibiting mitosis, and so they are keeping mitosis from happening in some respect. And so of course they're called tumor suppressors. There we go. Tumor suppressors, because the tumors form when that particular thing is missing. So if mitosis is missing, a tumor is going to form because it's going to start growing and growing and growing. Okay, the HPV is making proteins that will interfere with its own tumor suppressors. So normally your checkpoint genes are tumor suppressors. If they're in place, then you don't get a tumor, or it keeps it from growing if there is one. But in the case of HPV, the proteins that are making those checkpoints say, hey, let's not make this tumor, those are missing, so then a tumor will form. So tumor suppressors that make tumors form by stopping the mechanism that normally stops tumors in its place. So if you have a tumor, it doesn't necessarily mean that you're going to have cancer. Tumor, remember, is just a lump, a form of an area of cells that have grown out of control and formed a lump. And at some point, the tumor cells were suppressed and that lump stopped growing. That, in most cases, is called a benign neoplasm. And an example would be a mole. They're usually non-cancerous. If it didn't stop growing, or other tumors popped up elsewhere in your body and caused you sickness, then we would call that a malignant neoplasm. And of course, malignant tumors are life-threatening. Once they become life-threatening, we call them cancer cells. They occur when abnormally dividing cells of a malignant neoplasm disrupt your body tissues physically and metabolically. So, some tumors are good and some tumors are bad. And malignant neoplasms are important to recognize right away because they can break free and invade other tissues. And you've all heard of metastasis. That's when tumor cells or cancer spreads from one organ to another. This is a great pictorial of how metastasis works. So let's look at the picture. At number 1, you have abnormal growth of cells. So a checkpoint has gone awry at some point here, and you've got this tumor forming. Now at this point, the tumor could just be a lump and it stays there and nothing else happens, and it could be benign. Or it can be cancerous or malignant, and it will start weakening the cell membrane or cell walls, which is what number 2 is showing. And as it does so, cells are released and land on blood vessels, let's say. They can worm their way into blood vessels and flow to other areas of the body. Once it gets to an area that it likes, it will stop and worm its way back out of the blood cell into another area of the body. And so let's say it goes from the liver in number 1 to the lungs in number 4. And so cells will do this again and again, and that's how we get metastasis. Now, some cancers, depending on the type, will spread very quickly, and others will spread slowly. The ones that spread slowly give us time to actually radiate or give chemotherapy to and get rid of them and kill them, and that's the goal. Unfortunately, those therapeutic processes also lower your immune system, leaving you open to other health issues. Once you have metastasis to the lungs, oftentimes that's the last place they land, and so your body is completely riddled with cancer cells, and once it hits those lungs, it's often very hard to survive. Doesn't say people don't survive, but it is difficult to survive at that point. There are 3 characteristics of all cancer cells. 1, they grow and divide abnormally, which is what we've been showing you. And 2, they have abnormal plasma membranes, cytoskeletons, and chromosome number and metabolism. The abnormal plasma membrane and cytoskeleton are what make it weaker so that the cells can burst through that tumor membrane and move to the blood cells. Okay. And finally, number 3, cells do not stay anchored properly in tissues because plasma membrane adhesion proteins are defective or missing. So the certain types of proteins that can keep the cells adhered to one another in one spot, but usually they're defective or missing, and so that it weakens that plasma membrane, and then they can move freely throughout the body. These pictures are not very pleasant to look at, but they are pretty good ones to show you different types of skin cancers. The first one is a basal cell carcinoma. Basal cell is a type of tissue layer, so your tissue has many layers to it. The basal cell, I believe, is at the bottom layer, so this is a deep cancer cell. It's slow growing, maybe reddish brown. It's got sort of a shininess to it. Your second cell in the middle, those are the second common forms of skin cancer, squamous cell carcinoma. And again, carcinoma is a word that means cancerous tumor. Oma means tumor. Carcino means cancerous. Squamous cell means it's got one layer, it's one layer thick. So instead of having 4 or 5 layers deep, this is the top layer, it's squamous cell. And they're usually flat types of cells. These are pinkish in form, they're firm to the touch, and they grow under the surface of the skin. And of course, you've all heard of the last one, melanoma. Melanin is a pigment which creates this darkness, and there's a pigment or a melanin layer in your skin normally, which is what gives us our skin color. So the more melanin you have, the darker your skin. Melanoma is an abnormal tumor of that melanin. Okay, so they will spread the fastest. They're dark crustiness like this. They may itch or they'll bleed a lot because they pop open because they're crusty. These are incredibly difficult to get a hold of. So the second you notice something odd on your body happening, you must get it checked out immediately. Melanomas metastasize very quickly. So it's very important that you get that checked. Now, in the past, melanomas have always been sort of a death sentence, which is not true anymore. If it caught early enough, the tumor can be removed and you'll be given certain medications, not like radiation or chemo, but you'll be given some medication to help stop this from growing. Hopefully, once it was excised, all the margins were taken out and you had clean margins left. A lot of times, because they spread quickly, you'll have a large area of darkness by the time you notice it. And so if the whole thing can be excised, that's great. And oftentimes, though, you'll have to have a skin graft to replace that material because it was a large area. And so if caught early enough on any of these, a death sentence can be forgiven, if you will. You can avoid it. Now, I talked about clean margins. I don't have a slide for this, but if you look at the last picture, I think it's going to be easiest to describe. If you were excising this tumor, you would not follow the black edges exactly. You would go outward, maybe an eighth to a quarter of an inch, all the way around, getting some of that pink skin with the tumor. And what this does is hopefully you've gotten all the margins. Margins just means the outer edge of the tumor. Hopefully you've gotten all the margins of that tumor out. So now, if you're taking it out, and this is the only tumor in your body, and you're taking this whole thing out plus some extra healthier material, then all the cancer has been removed. And all that's left are clean margins, as we call it. Clean margins means that the tissue that remains that you have to sew back together, that tissue has no malignant cells in it at all. And so that's what clean margins are. And then if it was small enough area, you could sew it, suture it shut, but if not, then you'll have to have a skin graft. So clean margins is what you strive for if you're removing tumors. Now, sometimes what happens, there are tumor cells underneath this pink skin that have not yet pushed upward, so we don't see them. So even if we're taking a nice clean margin, we may not have gotten them all, and we don't know that until we have a pathologist at a lab look at it. So they would look at this under the microscope and they would see, yep, I see the margin of the tumor cells all the way around, and yes, I see a healthy circle surrounding it of healthy tissue. I do not see any malignant tissue in what appears to be healthy tissue. And so I would say that the doctor who excised this tumor got all the abnormal cells, and only healthy cells remain, and so they have a clean margin. And so then we're good to go. And nothing more needs to be done with it. Now, a lot of times what happens is if they're cutting this open excuse me, if they've excised this and the pathologist is looking at it, he may see one or two or more malignant cells in that pink tissue and say, oh, no, they didn't quite get it. They need to go back in and remove more healthy tissue. And so, or what appears to be healthy tissue. So the doctor would bring you back in and they would cut another 1/8 inch around and send that off to the lab. And now hopefully they've gotten it all. So it can be kind of an annoying process. It's very common to have to come in twice for tumor excisions that are, especially for the melanomas.